All right, so now we're going to keep going with photosynthesis and break it down into its different components. So hopefully you watched the video that basically goes over an overview and you have seen the different pieces that we're going to be looking at. If not, stop and go back and watch that video first. So overall, what's going on in photosynthesis is that we've got two different sets of reactions that are designed to eventually make sugars, glucose. And from there, we can utilize that to make all of the other macromolecules that the plant would need. It's divided into two stages, which historically were called the light reactions and the dark reactions. You can see there's a big red arrow that says no, okay? A better terminology would be the light reactions and the light independent reactions. So stage one, the light reactions, relies on light. Plants can only go through this first stage when light is available as a reactant. The quote unquote dark reactions, when they were named because they didn't require light. But the terminology seems to make it sound as if the plants can only undergo this second stage in the dark, which is absolutely untrue. So a better terminology would be to call them the light independent reactions, meaning that they don't rely on light. Or you could just call it the Calvin cycle, since that's what's happening during the light independent reactions. So we have two different pieces. In the light reactions, we are undergoing an exergonic reaction. We're going to utilize light to make ATP. So we're going to have ATP synthase. And we're going to reduce an electron carrier called NADP. This is the part where oxygen is actually a byproduct. So the oxygen that we're breathing is a byproduct of the light reactions. The ATP and the NADPH that we're going to have made during the light reactions is going to be utilized in the Calvin cycle, or what's called the carbon fixation cycle. Here's where we're going to undergo a very endergonic reaction, and it's going to utilize pretty significant amounts of ATP and NADPH, and it's going to convert inorganic carbon in the form of carbon dioxide into sugar. Now, what do we think the plant's going to do with that sugar? Hopefully, you're going, I don't know. Maybe they would utilize sugar in the same way that we would, and they would then utilize it to make more ATP. If that was your answer, you're totally correct. Good job. Okay. Yeah, people tend to forget sometimes that plants also go through cellular respiration. So they don't just utilize photosynthesis because they really are not getting very much ATP. They're making some, but it's not a significant amount of ATP. They make sugar, which then feeds into the cellular respiration in the same way that we've already talked about. So they're going to utilize the sugars made, put them into the mitochondria, and get ATP out of it the same way that we do. So all of this is relying on chlorophyll. So this is a pigment that's found within the thylakoid membrane here. What we have is what's called the porphyrin ring. You can see it here. It's basically kind of like, it's usually described as a decentralized cloud of electrons that are able to be excited by certain wavelengths of light. Particularly, the photons here are going to absorb violet and red colors of light. Hence, they are reflecting back green, which is why we tend to see green plants. Like all things in nature, everything wants to come down to its lowest possible energy state. So when these photons become excited by the light energy, they want to get rid of it. Okay, they want to come back down to their lowest energy state. So they're trying to basically just throw this energy to something else. And that's what's going to be happening as that energy is bounced around in what are called photosystems. So photosystems are where light energy is converted to chemical energy in the form of a high energy electron that can be transferred from one thing to another. So what we have in a photosystem are a series of what are called antennae complexes. What we have in an antenna complex is a bunch of chlorophyll molecules. So they can interact with light, and again, when they become excited, they don't want to be excited. Unlike humans, most things in nature don't like excitement. They want to come down to their lowest possible energy state. So they bounce it around. It's almost, think of it like a ping pong ball. You know, it's hitting one of these and it's bouncing over here, and then it bounces over there, and then it bounces over here, and it bounces, until eventually it bounces to what's called the chlorophyll special pair in the reaction center. This special pair of chlorophylls basically hold the electrons at a lower energy state than the others have the ability to do. So when the energy is captured by that special pair, it's trapped. It can't be bounced any farther. So what we say is that the special pair accepts this energy. And that's where we're going to be able to convert something that started as light into a chemical energy that we're going to see is then going to be passed via a transferable electron. So overall, what we've got going on in these photosystems are chlorophyll molecules that can harvest light energy, bounce it around randomly until eventually it hits the reaction center where it's going to have that special chlorophyll. At that point, the energy that was in that light is trapped. Now what? What do we do with that trapped energy? In a reaction center, what happens is that the special pair passes that electron to an electron carrier. So we've seen electron carriers before. We saw ubiquinone, we saw cytochrome C, so we've seen them before. Similar idea here. When the special pair hands off that electron to the electron carrier, there's going to be a separation of charge. Because remember, the electron is negatively charged. So what happens is the special pair becomes positively charged because it lost the electron, and the electron carrier becomes negatively charged because it gained the electron. We now have a separation of charges, and at that point, we now have chemical energy. Now, what are we going to do with that chemical energy? Send it on down the line. Okay, we're going to move it into the photosystems where we can then utilize that chemical energy to do work. So we start in photosystem 2, not photosystem 1. It's important to realize that photosystem 2 comes first, photosystem 1 comes after. They were discovered in the wrong order, so they got named in the wrong order, but we start with photosystem 2. Here we're going to see something very similar to what we have already seen. The goal of photosystem 2 is to generate a proton motive force that ATP synthase can eventually use to make some ATP. Not a lot of ATP, but some ATP. So if we look at photosystem 2, we've got our antenna complex, and then we've got our reaction center. And we know that the chlorophyll is going to harvest this light. It's going to bounce around, and eventually it's going to hit that special pair in the reaction center. that point, that high energy electron is going to get transferred to our electron carrier, which in this case is called plastoquinone. It's very similar to ubiquinone. Okay, so this is going to laterally diffuse through the membrane, and it's going to deliver that electron to a protein that's going to function as a pump called cytochrome B6F complex. So this is almost at this point exactly what we saw in electron transport. We are pulling a high energy electron through these embedded proteins and using the energy that was in that electron to pump protons. Okay, so very similar in mechanism to what we've already seen. So we know there's similarities. What are the differences? Remember that in the chloroplast, we have a extra compartment. So we are pumping protons into the thylakoid space from the stroma. So this is not happening along the inner chloroplast membrane. This is happening along the thylakoid membrane. We also only have one proton pump, so we don't have a succession of proton pumps. But again, that's because photosynthesis is not the primary means of making ATP for the plant. Photosynthesis is the primary means of making sugar, which then we can utilize in cellular respiration. So if we look at photosystem 2 a little closer, looking at the reaction center, when the special pair loses its electron as it passes it to plastoquinone, it's going to have to be replaced from something. Remember when we were talking about cellular respiration and we talked about how the entire process would grind to a halt if we couldn't regenerate NAD+? We said everything would stop. We couldn't even do glycolysis without that. In your case study from the previous module, Module 6, you were studying the effects of cyanide on the electron transport chain, and essentially it stopped you from regenerating NAD, which eventually killed you. So this concept that everything has to be regenerated for these processes to continue is pretty much across the board. So here, our big issue is we have to replace the electron. Otherwise, photosynthesis will grind to a halt. So where are we getting the replacement electron in photosystem 2? This is where water is going to come in. So remember that water was a reactant here. So what's going to happen is we are going to split water. Okay, we've got a water splitting enzyme here. It's got a whole bunch of manganese in them, and it's going to allow us to split water and pull the electron off. The electrons will move into the special pair to be replacement electrons. The hydrogen are in that thylakoid space, which, I'm going to go back, we can see is great because we're going to be able to utilize that. So this is happening over here. So as we're splitting water, we're also contributing to that proton motive force. And what's left is molecular oxygen. So it takes 2 water molecules and you remove 4 different electrons, and eventually we end up with oxygen. This right here is the oxygen that we breathe. So the oxygen that we breathe is synthesized so that we can replace the electrons in photosystem 2. Now, what happens in photosystem 1? We've got a similar setup. So we've got the antenna complex, we've got the reaction center, we've got a special pair, and now we have a different electron carrier. We've got ferredoxin and a different enzyme. So we've got ferredoxin NADP reductase. still in the thylakoid membrane. Okay. So the goal of photosystem 2 was going to be to generate that proton motive force and get a little bit of ATP. The goal of photosystem 1 is to generate NADPH. So that is the goal of this photosystem. Our high energy electrons are going to be passed to ferredoxin, which is then going to pass those electrons to ferredoxin NADP reductase, which is going to utilize that to reduce NAD plus to NADPH. So here we have seen the reduction of electron carriers before. So this is all similar stuff. This is nothing new here. We just have a different carrier. Okay. So the goal here is going to be to synthesize NADPH. So if we put it together and kind of see, okay, how does photosystem 1 and photosystem 2 go together? We start here with photosystem 2. During that time, we're going to utilize light energy. We are going to pass the energy eventually to the special pair, okay, which will then pass the high energy electron to plastoquinone. Now we've lost the electron, so we have to replace it. Where are we going to replace our electron in photosystem 2? From water. That's going to release hydrogen into the thylakoid space and the oxygen that we breathe. That electron is going to be ferried across here using plastoquinone to the cytochrome B6F complex. That is a proton pump, and it is going to pump those protons into that thylakoid space, setting up that proton motive force that ATP synthase can use. Now, what happened to that electron? Okay, it was being pulled through here. Where did it go? The electron, as it gets pumped through or pulled through the cytochrome B6F complex, gets passed to a second carrier, which is called plasticyanin. Plasticyanin takes that electron to photosystem 1. So when photosystem 1 releases its electron to ferredoxin, it needs a replacement electron as well. Over here, we got the replacement from water. Over here in photosystem 1, the replacement electron comes from photosystem 2. It's this electron right here that gets pulled through and ends up replacing the electron in photosystem 1. The goal of photosystem 1 is to make NADPH. If you go back to the beginning, we said that the goal of the light reactions was to make ATP and NADPH. There's our ATP, and there is our NADPH. Now, maybe you've thought of this, maybe you haven't, okay? And if you haven't, no big deal. That's what I'm here for. So, in cellular respiration, we said that water had a very, very positive redox potential. In other words, it had a very high affinity for its electrons. Remember that it was the final, oxygen was the final electron acceptor, and we made water, and water was not giving up those electrons. Okay, that's why it was at the bottom of the electron transport chain. And yet, here we are starting with water, which has a very high positive redox potential. It doesn't really want to get rid of those electrons, which means that this is going to require some significant pushing of these electrons, which we call boosting. That's what happens when the charges are separated in that reaction center. So we start down at, close to 1200, and we have to boost that electron up pretty high, and by up, it's obviously down, so to a negative redox potential. So we boost it up to about negative 600. Then plastoquinone is below that. So plastoquinone has a higher redox potential than here. And then similarly, we keep going down through the cytochrome B6F complex and then to plastocyanin. So we require this boost so that we can then go downhill from there. What's giving us the boost? That's the light. Okay, that's where the light is actually coming in, and that energy from the sunlight is boosting us high enough that we can then move down the redox potential scale. When that electron from PS2 gets moved over to plastocyanin, it comes over to that second reaction center in photosystem 1 and requires more light to boost it up so that it can go downhill to ferredoxin and then further downhill to our enzyme complex. So the light is actually giving us the energy to get these electrons to lower redox potentials so that they can then move downhill through these processes. So we've made the NADPH and we've made the ATP. What are we going to do now?